Thanks again for your kind invitation. This is really a rehash of data that was presented at the ACC and published in Circulation earlier this year. My disclosures, I was an investigator in the Chocolate Touch trial. This was what Jay was expressing in words earlier, that the FemPop is a torture test for all kinds of technologies. And then when it comes to drug elution, we now have a menu of options. But I think it's important to review the basics, and I talked about this in the fellows course yesterday. We have drug-coated stents. That's what PK is talking about with the Zilver PTX. We have drug-eluting stents with a durable polymer that will deliver drug over a period of time in a controlled fashion. We have drug-eluting balloons that will be forthcoming soon in terms of clinical trials. And then the conventional drug therapy we have is drug-coated balloons that leave an excipient and drug together on the inner lining of the vessel. In every case, though, you have luminal delivery of drug. It diffuses across the vessel wall, and the concentration of drug over time is what's proportionate to its effect. So it doesn't matter how you get the drug there as long as there's enough for long enough to have therapeutic efficacy. So the typical issue is with drug-coated balloon, you leave a high payload of drug, and then very rapidly it tends to decay away until it's not detectable. The area under this curve is what dictates its clinical efficacy. With a drug-eluting stent, on the other hand, you're able to provide scaffold support and also a slow and steady release of drug. But nevertheless, the area under this curve is also what dictates the therapeutic efficacy. So one thing that has been sort of lost in the weeds a little bit is that the drug-coated balloon technologies, even if using the same drug, are not all the same. Using that paradigm I just laid out, these are clinical data or preclinical data from our lab at CRF from my partner Juan Granada. This is using 3 commercially available DCBs in a pig model, and I'll walk you through the data. If you use Lutonix, Impact Admiral, or Ranger, these have different doses of drug and different excipients. What you find in the tissue is that you have differential drug retention. At first, you have a very high concentration of drug, but with some balloons like the Lutonix, for example, you have a relative rapid decay of that concentration. With other drugs, with other drug and excipient compounds, you have a slower decay. Now, interestingly, the Ranger has roughly half of the dose of the Impact Admiral. But nevertheless, you see that the tissue concentrations are very similar, and this is recapitulated in histopathology and in other markers of inflammation and thrombus formation. And so it's not surprising. The Impact Admiral has, I would argue, is the leader in terms of long-term follow-up and data and has excellent long-term patency. And it's largely because of this ability to retain drug long enough. So in this milieu, we now have new drug-coated balloons. The Ranger, obviously, as you would have predicted from the preclinical animal model, has similar drug retention, and therefore, in a direct head-to-head randomized trial, it has similar freedom from clinically driven TLR, similar patency with bailout stenting in short lesions, midterm lesions, and long lesions. So it's really about keeping the drug in the artery. So in this context, now the chocolate touch balloon, I think everybody's familiar with the chocolate balloon, which is a nitinol encaged balloon that has a protrusion of the balloon within the interstices of the cage of nitinol that allows for a pillow effect of vessel apposition with the balloon. This balloon, when coated with a combination of an excipient with a paclitaxel, has been then developed in terms of a DCB technology, which would have the advantage of being extremely non-compliant and therefore would perhaps save you a step in lesion preparation. The chocolate touch DCB was subsequently developed with a propyl gallate excipient compared to the polysorbate sorbitol excipient of the Lutonix device, and was developed with a slightly higher dose. And then the design of the study was to have a randomized controlled study comparing 1-to-1 chocolate touch to Lutonix as a predicate device, randomized throughout the United States, Europe, and New Zealand between 2017 and 2020. There was a series of follow-up patients, follow-up schedules set up with outcome measures assessed out to 5 years with the primary effectiveness endpoint of DCB success, which was defined as a composite of primary patency in the absence of a clinically driven bailout stent. The safety endpoints were freedom from major adverse events at 12 months with a composite of target limb-related death amputation, or clinically driven TLR. The primary efficacy endpoint was a non-inferiority assessment, as was the safety, and then there was a sequential superiority statistical analysis plan that was set up a priori to assess trial success. This was led by Mehdi Shishapur, University Hospitals Cleveland Medical Center, and Thomas Zeller in Germany, with core lab at Yale with Alexandra Lansky, and with a duplex core lab in Germany with Dr. Zeller. The overall study enrolled 313 patients, 152 in the chocolate touch arm and 161 in the Lutonix arm, and we had 94% roughly follow-up out to 12 months. The age and baseline characteristics were well matched. There's similar complement of risk factors in this patient population. Notice that there was predominantly Rutherford III intermittent claudication with a burden of diabetes of approximately 40%, with a healthy number of patients with multivascular disease and chronic kidney disease with a baseline resting ABI of 0.7. The lesion length in the study, as with most pivotal DCB trials, is relatively short, approximately 8 centimeters in both groups, with only about a quarter total occlusions and a quarter with severe calcification. In contradistinction to other DCB trials, atherectomy was added as an amendment later into the study, and approximately 1/8 of patients in both groups were treated with atherectomy. The amount of dissection requiring bailout stenting with flow-limiting dissection, and this is not a typographical error, it was in fact 0. I know that the core lab has spent a lot of time because nobody believes when you have a 0% complication rate, but this was in fact the case. This is the primary efficacy endpoint. You can see that the 1-year Kaplan-Meier evaluation demonstrates a primary efficacy endpoint of 83% with the chocolate touch balloon versus 73%. And the analysis inclusive of 13 months was 78.8 versus 67.7 with a P of non-inferiority that was highly statistically significant. The forest plots looking at virtually every other subgroup analysis demonstrates similar trends in all groups, in men and women, US and OUS, diabetics, those with greater than Rutherford III, those who were treated with atherectomy or without, with a trend towards perhaps greater benefit in the atherectomy group, in long and short lesions, and in differential amounts of calcification. So the primary safety endpoint was also met with a primary safety of approximately 89% in both groups, with a non-inferiority p-value that was highly statistically significant. So this was, then there was no mortality difference. This was obviously both paclitaxel drug-coated balloons. And mind you, this was done into the COVID-19 pandemic. And so there were no changes, no differences there. So in conclusion about the study demonstrated that the chocolate touch balloon met its primary efficacy endpoint with DCB success. It was both non-inferior and in sequential statistical analysis superior. It also was non-inferior for safety, and there was no mortality difference. And these data were published in Circulation. So I think, getting back and germane to this case, we have a menu of options for the treatment of the SFA. This study adds that differential drug-coated balloon technologies can be assessed head-to-head, and there are differential efficacies between them. So some balloons may work better than others. And there may be differential ability to distinguish these technologies when adding or layering on other therapies, such as atherectomy or scoring balloon technology. Our current appropriate use criteria suggest that drug-eluting technology should be used as definitive therapy in virtually every scenario, and I would probably stand by that. And with these technologies, as you've seen already, we've really flattened the length to restenosis relationship such that we can expect surgical-like results in a case such as the one PK is showing. So everybody can come up with their own algorithm. This is mine. I think PK is demonstrating in this case that he's got his own algorithm in mind, and I look forward to seeing how he finishes the case. Thanks very much.